So thank you very much for the invitation. Thanks, Alan, for allowing me to give a brief overview of the research that I do. And today's talk, I've just titled it, How Our Environment Can Affect Our Genome and Influence Our Health. I'm trying not to make it very technical and try to just have a bit of more simpler research tools, methods, etc. in just to give you an idea of what we're doing. And this is what I do. I actually did my PhD and my postdoc in Munich. And as you can see, we're working really hard up there. In 2015, I moved to Brisbane. And that's the Oktoberfest in Brisbane. So I try to keep some of my environment constant because that's very important, as you know. And a bit of my background, as Alan has pointed out, I am a geneticist. So I've actually done my undergrad, my master's in England in human genetics. And then I was very interested in psychiatric disorders, stress-related disorders, and in statistics. So then over the last 7-8 years, I've actually moved to what I call the dark side of statistics. So I'm now a biostatistician, and I work on data analysis. And so I'm a senior research fellow at QUT, and I also used to be previously at UQ, and I also have some projects at QIMR, so I have a visiting position at these universities. So really just an overview of what I do and my research focus. I really want to understand how genetic and environmental factors interact together and how this can drive our health in a particular direction. And over the last few years, there have been a lot of different genomic technologies that have come up, and now we can measure the influence of the environment on our genome. So here, you would probably remember this from school. This is what we call the central dogma of biology. It's the flow of genetic information. So you have the RNA, DNA, RNA, protein, and then health. So at the level of the RNA, this is the gene expression. So we can measure the expression of genes, which is basically the activity of genes, how the gene functions. And the DNA here is made up of the 4 base pairs, A, G, C, T. And the DNA code is what we're born with, so that generally does not change. But what's interesting is that in our DNA, we have some sites. So these are sites in the DNA where the C and the G base pairs are next to each other. And these sites in our DNA are extremely sensitive to environmental factors. So due to several environmental factors, we can have chemical bonds, such as methyl bonds, that can be added onto our DNA. And this addition of the methyl bonds is known as DNA methylation. Now, DNA methylation is one of the most common and widely looked at types of epigenetic changes. So epi in Greek means on top, and epigenetics basically involves chemical modification of our DNA. So as I mentioned, the code does not change, but what changes is the expression, downstream expression, which means that through DNA methylation or through epigenetics in general, our gene function, gene activity can change, and this can influence our health. So epigenetics is a very new field. It's only in the last 7 or 8 years that we can actually look at a person's entire genome through this. On the top, you can see it's a microarray. So a microarray is really as big as a microscopic slide. You can take blood, saliva, buccal swabs, any kind of biological sample from a person, and you can just run, and you can look at a person's DNA methylation across the entire genome. So that's the kind of technology that we're using right now. Now, when we talk about epigenetics, we always think about a lot of different negative environmental factors, and a lot of my work, of course, has been related to stress. But there's also a lot of other positive environmental factors. So there's things such as exercise, which I'll talk about a little later, but there's diet, so diet can be important. Social interaction, social support, that's a positive environmental factor. So there's a range of positive as well as negative factors that can influence our epigenome and can influence the activity of our genes. So when I moved to QUT about 4 years ago, I started working together with our group on the Vietnam Veterans Project. And this is a study that was funded by the RSL Queensland together with our research partners, the Gallipoli Medical Research Foundation. This is a foundation based in Greenslopes. So we had 300 veterans that visited us, and 150 had PTSD and 150 veterans without PTSD. We've taken biological samples from the veterans. We've taken blood, but we've also taken sperm. And I'm not going to talk much about the sperm, but that's an interesting study and a talk by itself. And we've run these samples on the arrays that I've spoken about. So we've got DNA methylation for all of the veterans across the entire genome, but we've also got a lot of different types of data. So this is, in green slopes, this is where the veterans come for treatment. So they see a lot of psychologists, psychiatrists, physicians. So we've got a lot of different types of information available. And we have different PhD students who are working on different aspects of the veterans' mental health and physical health, actually. So for this talk, I just want to talk a little bit about what we found with the epigenetics, so the DNA methylation changes. And one way, as I mentioned, you can look at the epigenetics or the methylation changes is just, scan a person's genome and compare veterans with and without PTSD, and you can try and identify genes associated with PTSD. So we did that a few years ago, and for those of you who are not familiar, this is the Manhattan plot. So you have your chromosomes from 1 to 22, you have a p-value, but this is the minus log p-value, which means the higher it is, the more significant it is. And when we looked at veterans with and without PTSD and we compared them, we identified 5 genes associated with PTSD. And what was interesting is the DOC2 gene, we could replicate it in another sample, and this is a gene previously associated with Alzheimer's disorder. So this was quite interesting because a lot of the veterans we know who have PTSD also have different types of dementia and other neurodegenerative disorders. So it just tells us that there could be common genes that are involved in PTSD or stress-related disorders and other types of illnesses as well. But what's interesting is instead of looking at the specific gene, so each dot here, there is a way to actually look at an integrated value of this. And in 2013, Steve Horvath, he's an American researcher, he came up with this Epigenetic Age Predictor. So instead of looking at everyone's individual genes, you can get a single measure of a person's epigenetic changes across the genome, and we call this a person's epigenetic or their biological age. So this was quite interesting, and I wanted to show you a little bit about what this is. So this is a study we did together with my collaborators in Germany, and this is a plot of chronological age and your predicted epigenetic age. So if I come to you and ask you how old are you, that's your true age or your chronological age. And if I take your biological sample and I do this prediction, then I can actually tell you what your epigenetic age is. And as you can see, there's a very high correlation. So in general, most of the people's true age and the predicted epigenetic age is quite highly correlated. But you see a lot of outliers. And here in red, you see this is a person who's actually 20 years old, but is actually predicted to be 45 years old. So this is really interesting. And so the difference between a person's true age and the predicted epigenetic age is what we call accelerated aging. So in this study, we wanted to find out what is this? So why is a person who's 20 years old biologically, why is this person 45 years old? And we tested different measures, different things, and we found that accelerated epigenetic aging is associated with a person's cumulative lifetime stress. So this was interesting because we looked at recent stress, because a lot of people say recent stressors are important. We did not find this with recent stresses. It's actually an addition of the stress that you accumulate throughout your life that is important. And so the cumulative lifetime stress that influences your epigenetic age. So what does this mean? This basically means that we are all as old, so biologically or epigenetically as old as how stressed we are. And I love giving this talk because often everyone's really interested in epigenetic aging, and a lot of people say, oh, can you test my epigenetic age? So everyone wants to know their epigenetic age. Now, as you can imagine, a lot of people have used the Epigenetic Age Predictor. People have looked at it not just in psychiatric disorders, but in other disorders, so cancer, Downs, obesity, diabetes. And overall, the consensus was that accelerated epigenetic aging is a bad thing. So it's associated with a lot of different negative health outcomes. But no one's looked at the positive aspects. So we wanted to look at resilience. We wanted to see how does resilience look and what is the actual link between resilience and epigenetic aging in the veterans. And we looked at resilience via the Connor-Davidson resilience scale. So I'm not sure how familiar you are with that, but it's basically a questionnaire of 25 questions, and then you sum it up and you get a person's resilience scores. So as you can imagine, resilience is a good thing, we all agree, and epigenetic aging, everyone's shown, is a negative thing, so we expected a negative correlation between the two. And this is what we find. So this is the resilience here and the epigenetic age acceleration. So if you look at veterans with no PTSD, this is the blue, you see there's a negative correlation. So if you don't have PTSD, your increased resilience is associated with decreased age acceleration, which is a great thing. But what happens if you do have PTSD? And these are veterans with PTSD. So if you have PTSD, in the veterans we've seen that increased resilience is a bad thing because increased resilience is associated with increased epigenetic aging. So this was really kind of, we didn't expect this, and this really stumped us, and we had to think a little bit more about what this actually means. Now, as I mentioned, the way we calculated resilience was through the questionnaire. So there are 25 questions, and of course, one main question is, could it be that there are a few questions, could it be a subset of questions that are driving this kind of interaction? So we looked deeper into the questionnaire, and we did a factor analysis to see if there were specific parts of the questionnaire driving this unexpected result. And what we found was there were 9 questions, so here in red. And if you look back at the literature, a lot of psychologists and researchers have termed these as persistence-related questions. And it was these 9 questions that actually drove the unexpected results. And now let's look a little bit deeper into it. So you have things such as be a strong person, do not give up, put your best effort forward, do not be discouraged. Do you focus and think clearly? So these kind of things. And we had a really good chat with, with some of the clinicians who treat veterans with PTSD, and they told us this actually makes a lot of sense because if veterans are suffering from PTSD, these kind of thoughts and ideas are actually really harmful for them because there's often things that they cannot do routinely that they could do before they got PTSD. So certain things, so I guess under normal circumstances, resilience is a good thing, but if you are suffering from PTSD or depression, then certain aspects of resilience that we measure you're just kind of putting an additional toll on your own body. So it's kind of, the veteran said, yes, so this is important to know that we're not kind of forcing ourselves to do things that we shouldn't be doing. So these kind of persistent related factors we think are resulting in the cognitive, emotional, and a physical toll on the body. And that led us to a paper which we called the biological cost for soldering on, because we thought that was a really good way to put it. We've looked a little bit at exercise, which I think is interesting for this talk. We've looked specifically at the Bdnf gene. This gene is an important gene involved in neuroplasticity, cognition, as well as memory. And there have been previous studies that have shown, so in mice as well as in humans, that the Bdnf gene expression as well as the methylation can be altered via antidepressants, but also other different treatments. So we looked at the veterans, and we classified them. So these are older veterans. So of course, active exercise in people who are 60 years and older is slightly different than what we think active exercise is for younger people. And we classified them as active exercise and non-active exercise. And we found that so exercise was associated with changes in DNA methylation in the veteran. And these are 3 sites in the Bdnf gene that showed significant methylation changes. So a decreased DNA methylation of the Bdnf gene causes an increase in the expression of that gene, and that could act as an important therapeutic intervention. So this is quite important, and Bdnf is one of the kind of key genes involved in a lot of different processes. So not just psychiatric disorders, but a lot of memory, cognition, etc. So this could be a very interesting gene to look at. Now, I just want to stop here and say, so most of the studies we've done so far are cross-sectional, which means we've just looked at one time point. And one issue there is, of course, you cannot distinguish whether the changes we observe are a cause or a consequence of the disorder, because, of course, we did not take blood from the veterans before they went into combat, which would be great. So some of these studies have their limitations, and the other limitation being that we've often focused on the negative aspects of health. So we've looked at depression, PTSD, anxiety, etc. So this doesn't really give us a whole picture of health and well-being. And as Alan pointed out, I'm in the School of Psychology and Counseling, and at the school I met a professor who's very interested in looking at resilience and post-traumatic growth. So these are the positive aspects of a person undergoing a traumatic experience or having a stressor. So I joined together with Professor Jane Shakespeare Finch at QUT, and we started this new project where we're interested in looking at psychological and epigenetic changes that occur after exposure to a traumatic or highly stressful event in paramedic students. So we collected biological samples and a survey data from paramedical students at QUT and at USQ before and after their first field response exposure. So that's kind of one of the main stressful events that they go out to. So we've got basically epigenetic changes, DNA methylation changes before and after a stressor. And that's given us some interesting results. I just want to point out, of course, this is one of the first studies that has looked at the whole range of post-trauma responses, so kind of encompassing the positive as well as the negative aspects. And we've really, so we've just got a paper, so our students just got a paper published this week, and we found some key genes that are related with response to trauma. And also we find that there are different genes and different biological pathways that are involved in PTSD, post-traumatic growth, and resilience. So showing that there are different pathways and mechanisms that kind of can cause these differences in post-trauma trajectories. So this is what I thought would be really interesting with the athletes, and just a proposal. So as I've pointed out, we've shown that epigenetic aging plays a key role in health and well-being. There are some key genes involved, such as the Bdnf gene, where we've shown that exercise specifically can alter the methylation. So I would really be interested in looking at what the biological effects of physical exercise and training load are in athletes. I want to look at the changes in the mental and the cognitive performances in athletes before and after physical training. This would be quite interesting. And also try to look at different types of training interventions, looking at the cumulative training load, so kind of similar to what we've looked at with the epigenetic aging, and seeing how these differ and how they can change depending on differences in the interventions or the training. And I guess the way to go about it is, of course, to collect a biological sample. So this could be, buccal swab, saliva. Saliva is really easy. We've done it with the paramedics. You just get a tube and you just spit into it. It's disgusting, but it's really easy. And of course, I'm also interested in looking at immune-related genes, because immunity, immune genes play a key role in stress or physical activity and responses to physical activity. So it could be interesting to also measure some immune biomarkers. We've had so a PhD student, Dogma, who's worked with us, has done a bit of work in the veterans and looked at specific immune-related genes. So we've looked at the TNF-alpha and the IL-6 expression and methylation associated with PTSD and resilience in the veterans. So it could be interesting to look at some of these immune markers as well. And now I'm going to switch gears a bit and talk a little bit about hormones and postpartum depression. That's my second field that I'm interested in. So that's another field of research that I'm very passionate about. So all of us know that hormones play a very important role in the health of women, and for women, it's been seen that there are several negative health outcomes that happen during times when the hormones fluctuate a lot. So these are things such as pregnancy, but also menstruation, as well as menopause. And in athletes as well, hormones play a very key role because they're associated with fatigue and also reduced performance. So it's really important to understand the roles of hormones and how these hormones can influence our methylation or gene expression and also influence our health. So I guess my my start to this was through postpartum depression. So I've been very interested in postpartum depression, which is one of the main kind of female-related kind of disorders, which is caused by changes in hormone levels. And from the psychiatric point of view, it's one of the very severe disorder, because it affects not just the mom, but also the child and the family, and it's a community-wide issue. So if any one of you know someone suffering from postpartum depression, you'd know how bad the outcomes are for everyone. And one of the issues with postpartum depression is that by the time we detect it, it's too late. So usually the symptoms have manifested, and it's too late. So one of my main research focus was trying to identify early predictors of postpartum depression. So when I was in Germany, we did this study where we identified the earliest gene expression predictor for postpartum depression. So we identified a set of 116 transcripts, and if you look at the gene expression of these transcripts during the 3rd pregnancy trimester for women, we could actually build a predictor and predict with an 88% accuracy which women would go on to develop postpartum depression. So this was quite exciting for us, and of course we have replicated this, and I'll show you a bit later, but there are other groups replicating this predictor as well, because that's got a lot of clinical utility. But the way we found the 116 transcripts was we just did a genome-wide scan, so we didn't really have any specific candidates, we didn't have any hypothesis. When we looked at the 116 candidates of these genes, and we tried to find out what are these genes, what do they do, it was interesting that several different analyses all pointed towards the role of estrogen or the estrogen signaling pathway in these genes. So that was kind of really interesting that, we just ended up with estrogen. And I guess we wanted to find out, so how could estrogen play a role in postpartum depression? And there are 2 possibilities. So one possibility is that it could be that the women with and without postpartum depression, so the high-risk women, there are absolute changes in hormone levels, and this could be associated with postpartum depression. So we checked that, and we did not find that. So in fact, the absolute levels of the estradiol and estriol were not different between the cases and the controls. Now, there's an alternate explanation. So many years ago, Mickey Block and several other people had proposed what they call the estrogen receptor sensitivity hypothesis. So they did some experiments where they give women some hormones and they took it out, and they found that there were some women, when they took the hormones back, they suddenly had depressive symptoms. So they called it this kind of estrogen sensitivity, where they mentioned that there are a subset of women who are highly susceptible to these fluctuations in hormones. And it's this subset of women that are at high risk for hormone-related disorders and negative health outcomes. So we wanted to look at that. So we had the gene expression from women before pregnancy, so 0 here is preconception, and through postpartum. And we wanted to actually check, could it be that the gene expression profiles of these 116 genes, and we also looked at a set of estrogen responsive genes, do these look different among the PPD, so women with postpartum depression versus the controls? And if you can look at the graphs here, so this is the gene expression of just 2 genes here, and in blue you have the control women, and in red you have women with postpartum depression. You can see that the gene expression in the control women is quite controlled, actually, so it's kind of really, very similar. But if you look at the women with postpartum depression, they have a very dynamic changes in gene expression. So, kind of really pointing us towards the fact that, it kind of works more towards the estrogen receptor sensitivity theory. So just the conclusion from the study was there are a subset of women who are more sensitive to the normal hormonal cycling, and these women are more vulnerable to postpartum depression, but also to other hormone-related disorders. But we wanted to look at a larger sample, and of course wanted actual molecular evidence that estrogen signaling plays a role. And that's where we came to this collaborative work. And I love showing this photo. I'm a cat person, so I just really like this photo. So I presented our postpartum depression work in Europe at one of the conferences, one of the perinatal and infant health conferences, and got in touch with one of the Danish collaborators. And Wiebe here, she was working on a very similar theory, but through a very different model. So they did a clinical trial where they gave women a gonadotrophin-releasing hormone agonist, or the GnRHA. And I don't want to get too much into the details, but just briefly. So what the GnRHA does is it's got a biphasic response to hormones. So you have this increase in hormones and then sudden decrease in hormones, and that's due to this negative feedback loop that happens. So it's very similar. If you look at the hormone patterns, it's very similar to what happens in women late pregnancy, early postpartum. So this was a very interesting model to study for us. And in the same model, they found that this kind of changes in hormone cause depression in the women. So here in red you can see, and blue is the placebo. And this depression was specifically associated with the estradiol levels, and that's in the middle here. And even more interesting is that these changes were associated with the serotonin binding in the brain. So serotonin is a neurotransmitter, and there is some kind of there is a hypothesis that estrogen kind of affects our health and our mood via serotonin transporter and signaling. So we wanted to test our genes in this model, and we had longitudinal data. So we had gene expression and genome-wide DNA methylation data at different time points, and we basically did similar analysis, just testing if the genes that we found are different in the women who got this intervention of the GnRHA versus not. And that's we've published this. I just talked briefly going on to the results. So we found that when we looked at our panel of genes, and we also looked genome-wide, we found that there were gene expression and DNA methylation patterns in the women who had been treated with the GnRHA that predicted estradiol levels, but also depression, as well as the serotonin binding in the brain. So kind of just, in a way, giving us some kind of hope and replication of what we have seen in our postpartum depression sample, but here in a specific model where we know that estradiol plays a role. And it causes depression and affects brain signaling. So just the key findings here. So I guess both these studies, put together, just strengthened our hypothesis that there is estrogen sensitivity that plays an important role in postpartum depression. And I think this has a lot of different implications for prevention. So this is what I wanted to do. So I know that oral contraceptives and female health is very important in athletes. And the question here is there a subset of female athletes that show the so-called estrogen sensitivity? And if so, does this actually influence performance? So I think that's a very important factor. As all of you know, so oral contraceptives, most of them are a mixture of estrogen, progesterone, and they're different ratios. So this is kind of really, you're ingesting estrogen, essentially. So does that actually cause the hormone fluctuation? And are there some athletes that are more kind of sensitive to this hormone fluctuation? And I guess a more broader question is about optimal. So can we actually look at the gene expression and DNA methylation profiles, and can we actually determine and predict optimal times for training, performance gains? So training-induced psychological problems, look at the general fatigue that occurs after the ingestion of the oral contraceptives, and also look at different conditions, so high training load versus low training loads, etc. And that's just one thought. Similar methods, collecting samples, biological samples, survey data, and trying to analyze all these together. So just to kind of conclude, epigenetics is a very interesting field of research because it allows us to measure the influence of different environmental factors, also things like physical activity, diet, stress, other things on our genome. What's very interesting about epigenetics, and I think it makes it different from genetics, as I mentioned, the DNA code is what we're born with, that we cannot change, but we can actually change our own epigenome. So if we can change environmental factors, lifestyle factors, we can actually change our epigenetics and our gene expression, and therefore the activity of and the functioning of genes, and obviously change our health. And I guess the way I'd like to look at it is take a very multidisciplinary approach. Get a holistic view, as I mentioned, not just the positive but also the negative and the positive. So just understand completely what's happening and how different environmental factors, things such as exercise, but other interventions as well, can influence our epigenome and influence our health and our well-being. And finally, I just want to thank everyone. So top here, this is the team involved with the Vietnam veteran study. Jane and Dogma involved in the paramedic study, and Daggy also worked on the veteran study. And Elizabeth and Weber, these are my European collaborators. And I've got a grant from the NARCET to look at the postpartum depression predictor in a larger sample of American women. And that's together with Samantha Melzer-Brody and Naomi Ray at UQ. So thank you very much for your attention.